All right, so we're starting, Dedi. Okay, welcome everyone, and thank you for joining us for our sixth webinar, where we give you the opportunity to peek into our scientists' lab and get a better feel to and understanding to their research. We're very privileged today to welcome Dr. Dedi Meiri, who is probably among the world's top scientists dedicated to medical cannabis. He'll be formally introduced by our moderator today, Adam Miller. We're live on Zoom and Facebook, and as always, you're encouraged and welcome to ask questions. You can either ask directly on the Zoom, on the chat, or the Q&A section, or on Facebook, and we'll feed Dedi with your questions, and hopefully he'll be able to answer. And if not, at the very least, he'll look into it and get back to you like every good lecturer does. Before we dive in, my name is Ori Danieli. I am the Executive Director at Technion Australia. Technion, for those of you who don't know, is Israel's Institute of Technology. It's also among the world's leading institutions dedicated to science, technology, and engineering. And in Australia specifically, we're part of a global network of about 25 associations worldwide with a mission to support the Technions, both support our scientists, our research, and support from a philanthropic point of view for infrastructure, scholarships, etc. We also aim to strengthen and build on research collaboration. Finally, I just want to quickly thank our partners today. That's North Sydney Innovation Network, Jonah Herman, who's the founder and chair, which we'll hear from a little later on. He's been with us from the start. Thank you, Jonah. And the Embassy of Israel in Canberra. I particularly want to acknowledge our ambassador, Mark Sofer, who has been a long-time supporter of Technion. And Ron Gelstenfeld, who is the Deputy Chief of Mission, which we'll hear from in a minute. So Ron joined the Foreign Ministry in 2008. He has been a Deputy Chief of Mission in Uruguay between 2010 and 2015. And in 2018, he joined the embassy here in Australia as a Deputy Chief of Mission. So Ron, over to you to formally welcome our guests. Thank you.
Well, thank you, Ori. Thank you, everybody. First of all, thank you, Ori, for inviting me. I would like to mention that I have to, first of all, to congratulate you and Technion Australia for these so important activities that you're doing, both in quantity and in quality recently, in such a special circumstances that we live in. This webinar are really, really important. And this is what brings the word of Technion and the word of Israel to the public, both in the Jewish community and the general public, which is very important, the academic world as well. I do believe that this webinar will be one of the most attractive and interesting one, because when we speak about cannabis and medical cannabis, this attracts our mind and imagination, what can be done, and we hear so much about it recently. In the last few years, and the industry, such an amazing development. And thank you for bringing the distinguished keynote speakers today to bring us the word of Technion and research. Myself, as you mentioned, I was serving my previous posting in Uruguay, the first country in the world that actually legislated cannabis for recreational usage. I think the first one and maybe the only one so far, but the cannabis diplomacy was always an important part of the job that I did over there, and hopefully to be able to do also here in Australia. I must say that it's on and again, whenever we speak with government agencies, with academy and with industry, mainly when we're dealing with bringing them to Israel as part of a delegation, the issue of the medical cannabis is always probably one of the first issue of interest for Australia. I remember that I wasn't in Australia yet, but when Premier Daniel Andrews came in 2017 to Israel, was his main topic of interest, as well as Premier Mike Baird when he came here from New South Wales in 2016, and even in the planned visit of Premier of New South Wales, Gladys Berejiklian, that was supposed to be exercising last April, unfortunately for the corona, was not able to. This is what mainly she spoke about. I want to see the greatness of Israel research, industry, and knowledge regarding the medical cannabis, which give us a source of pride that even in this niche sector, we are world leader as much as Australia does. So thank you very much for allowing me, and Dr. Meiri and Adam, as I mentioned previously, you're going to hear from me pretty soon because I have a lot of ideas with the moderation of Ori, of course, and Technion for more collaboration, deepening the collaboration. And there's a lot of things to do. And again, greetings for the activity. Thank you very much.
Thank you, Ron. Appreciate that. I now want to introduce our moderator today, Adam Miller. Adam spent over 8 years building B2B businesses within education, investment, and medical cannabis. He also consults to private and listed cannabis producers and ancillary businesses. He founded and currently sits on the board of the Medical Cannabis Council, which is Australia's largest not-for-profit industry body. And he regularly appears in the media on areas relating to medical cannabis, which makes me very happy that you're moderating the discussion today. His weekly newsletter has over 20,000 subscribers, which we encourage you to sign up. We'll send you a link later on. So on that note, Adam, over to you. Thank you.
Thank you very much. I appreciate that, Ori and Ron. Thank you for the introduction. Everyone, thank you so much for making the time to join us today. I can guarantee you, you're not going to be disappointed. Dedi is, in my view, the most talented researcher on the planet when it comes to medical cannabis. And I don't say that lightly whatsoever. So just a little bit of background to Dedi. Dedi is an associate professor at the Faculty of Biology at the Technion and a member of the Technion Integrated Cancer Center. Dedi holds his master's in biochemistry and a PhD in plant biotechnology from Tel Aviv University. He conducted his postdoctoral fellowship at the Ontario Cancer Institute, where he focused on tumor invasion and metastasis. During his postdoctoral studies, he expanded his knowledge in human biology and cancer research and gained worldwide recognition as an expert in the field. Upon completion of his postdoctoral fellowship, Dedi took it upon himself to work at the Technion Institute of Technology, where he heads up the Laboratory of Cancer Biology and Cannabinoid Research. Presently, his lab investigates the therapeutic potential of phytocannabinoids with a focus on anti-tumor effects of cannabinoids. In addition to the laboratory and the cannabis research that he conducts on site, he also works with growers, clinicians, manufacturers, distributors, and most importantly, patients in his pursuit to understand how cannabis affects human physiology. I am deeply honored and very happy to be presenting Dedi. As I mentioned, he is no doubt one of the most talented people on the planet right now when it comes to medical cannabis research. So, Dedi, I'm going to hand over the mic to you. It's an absolute pleasure.
Thank you very much, Adam. Thank you very much, Ori and Ron. It's a big honor for me to be here. Thank you very much for all the audience that take the time to listen and to be here. I got mails and WhatsApp from all over the world that people are watching us, Australia, New Zealand, and other places. So it's a big honor for me to give this talk. I hope it's not what we call friends and family that hear me already 20 times, because the science is not changing in 1 week. So I hope I can show a few new things and mostly my view on the field and what we can do and achieve and which windows it's open in medicine in general. I will try to do a kind of a short talk, and then after that I will chat with Adam about the question that he have and question that we will get from the audience that we will answer. So I will just share my screen that we will be able, no, it's not this one. Oh, so hold a second. I'm always in this webinar. It's very funny because you're giving professor in the Technion to, so can you see my screen now?
Not yet, Dedi. Bottom of the screen.
I need to do share screen, and then you will be able. And why? Okay. This is the share screen, and we will be ready in a second. And you can see that now?
Yep.
Okay.
You can see it. There you go.
So again, to people that don't know me, I have a laboratory for cancer research in the Technion, and 5 years ago, I started to work with cannabis. My background, as Adam mentioned, I have a PhD in plant biology. I'm also a son of agriculture. With my father had a big farm. So plants are coming native to me. It's very easy. And when I got my lab in cancer research, I want to see how I can combine plant with my research. And there was a publication that showed that cannabis affect cancer, and I want to check the anti-cancer properties of cannabis. 5 years ago, it was kind of a desert, like we didn't know anything. There was 8 authorized growers in Israel that give cannabis to patients. In Israel, there is medical cannabis from 2007. So there was a little bit knowledge. There was around 50, 60 different types of cannabis. So I went to one of the growers, Shai from a Siach company, and I asked him few strains of cannabis. Of course, I got a license from the government before. And we started to work with that. And what we did, we took breast cancer cells and we grow them, and you can see them in this figure. And then when we took cannabis extract in very low doses, extremely low doses, it's killed these cells. It was very surprising. Every material will kill cancer cells eventually, but there is something we call IC50. What is the amount of the materials that we put on the cells to kill them? And we put an amount that is actually quite similar or even lower than the regular chemotherapies that killing these cells. And we were very surprised that in this amount, the cannabis killed these cells. And then we took extract number 2, which is a different profile of cannabinoids, which are the active compounds of cannabis. And even though we looked that days mainly on CBD and THC, this extract also killed the cells. But the third extract that was similar to extract number 2 by these 2 compounds didn't affect the cells. What we learned from that these 2 phytocannabinoids from the plant are not the only one that's active, and there is other compounds. Going deeper and deeper, we learned that there is a lot of active compounds in the cannabis plant. Actually, we have 3 major phytocompounds that affect our body. It's the cannabinoids, the terpenoids, and flavonoids. These are the major 3 families of compounds. The phytocannabinoids, these molecules that you see now, are actually being discovered by Professor Rafi Meshulam in year 64 in Weizmann Institute. Rafi then moved to Hebrew University and still active professor. He was 90 years old 3 months ago, but he's still active. And he discovered these molecules. And later on, he discovered that there is an endocannabinoid system in our body that actually produce these compounds, and our body have this machinery within that create these molecules and balancing the activity of our body. Almost every organ, every system in our body will have endocannabinoid system that balancing it, doing fine tune of how we sleep, how we eat, how we feel pain, how we feel appetite, our immune system. They're all being controlled by our endocannabinoid system. And this molecule that cannabis plant produces binding this system and actually change this balance. And this is the reason cannabis affecting so much. But we learned that it's not enough to look on these 2 phytocannabinoids, because the plant is producing many phytocannabinoids, also terpenoids and flavonoids, and our body produces a lot of endocannabinoids. So I understood quite fast, and in order to understand what is doing this effect on the cells, I need to develop method, first of all, to analyze this compound and to know what I have in my hands. Just for an example, today I have more than 900 different types of cannabis. Each one of them contain hundreds of different compounds. So it's a big variation. So we developed, first of all, method to analyze all the phytocannabinoids. Today we can analyze 10 different families of phytocannabinoids in the plants, altogether around 140 phytocannabinoids in the plant, different molecules that affect our body. We moved on and we learned how to analyze all the terpenoids, the other family. And again, today we can analyze from the plant around 150 different terpenoids. We moved on and we did the same from flavonoid. And then we moved to our body and we took blood samples, and from blood samples or from tissues, we can analyze around 150 endocannabinoids that our body produces. Making all this put my lab in a situation that we were kind of the only one that have this ability to analyze this variety or to look in this scale of how the plant is working. And it changed the lab totally. We started to work with other collaboration with physicians and growers. We started to look how it affects epilepsy, Alzheimer's, sleep disorder, multiple sclerosis, immune system on top of cancer. And I want to show one example how my lab methodology that we are taking, don't forget that English is not my first language, sorry for that. So how we're working and what we're doing. So when we're taking a type of disease, and it doesn't matter if it's multiple sclerosis, cancer cells, sleep disorder, or whatever, we're starting with a fast screen. We have in my lab equipment that can screen very fast a lot of samples. The equipment that you see now can take 1,000 images an hour. It's a fluorescence microscopy that can move between different samples, take images. So in 24 hours, you will have 24,000 images. And you can start to do statistically and other examination and to learn how many cells died, how many cells move, how many cells proliferate, or whatever you want to see. So from this equipment, we can see these results. Every color, it's a different type of, oh, I did this mistake again. I did it yesterday. Just a moment. Let me go out of that. And I need to remember how to do that. Hold a second. No, not this one. Okay, we're back. Sorry for that. I want to put a pointer, but I did something here, pointer. This is what I want to do. So every color, it's a different cannabis chemovar, different strain, different type of cannabis. And what we see leukemia cells that we're using on them cannabis, and we're looking on the survival, the viability of these cells. So if the line is lower, more cells are dying. You see that most of the cannabis strain that we use on these leukemia cells didn't affect the cells. But there is one buster, one strain that killed these specific types of leukemia very efficiently. And what we learned that these leukemia cells, these specific leukemia cells, have a specific mutation called Notch1 that causes 50% of the leukemia. It's caused in 50% of the cases of the leukemia, there is this mutation. So we took cells, leukemia cells that doesn't have this mutation, and we saw that the cannabis is not affecting them. But if you have the mutation, the cannabis is very affecting killing these cells. So we moved on and we did all the biochemistry and the cell biology. We understood exactly what the cannabis is doing, is inhibiting a protein in the cycle of this protein that in the Golgi, don't send it to the membrane. I don't want to go to details of the biochemistry, but we did everything that I know on cell biology, and we understood exactly the mechanism, how the cannabis is blocking this pathway of this mutation, and why cells that with this mutation are dying and normal cells are not dying. And we did everything. We took a normal cells, we create this mutation, we saw that they become sensitive. We take cells with the mutation, we correct the mutation, then become resistant. We did everything that I know as a cell biology and a cancer scientist to do to prove that what we see is real. Then when we move to mice model, we create these tumors and these specific mutation type of tumors in the mice. And in 3 different mice models, we show that if we're treating the mice that have these tumors with this extract for 14 days, we can reduce dramatically the size of the tumors, and you can see the stat here. And it doesn't matter if you are creating the tumors first, injecting them there, the size is reduced, or we started to inject the cannabis from the beginning and the tumors is not appearing. So it's kind of blocking metastasis and also killing the initial tumor. Because we're talking with leukemia, we did a bone marrow implantation and a radiation to the mice, and we create this leukemia, and we show that we also can cure this leukemia in the mice. So we did everything on the preclinical that I'm familiar to do with this specific cancer type. Then the $100 million question: do we need 1 molecule of this extract? Why is unique? Why other CBD strain not affecting these cells, and why these specific types of cannabis is unique? In 1998, Rafi Meshulam published a paper called the Entourage effect. Actually, this paper was talking about how endocannabinoids is activating in our body, and he showed that sometimes 1 endocannabinoid is not enough. You need few endocannabinoids in order to have an action. And it's called the Entourage effect. It's kind of synergism between the molecules. In the cannabis field, we took this idea to the cannabis, and we said, when you took in 1 molecule from the cannabis, it's not working as good as the whole extract. So in this case, I'm asking, do we need the whole extract? Do we need this Entourage effect? Or whether it's 1 molecule, 3 molecules, 7 molecules, I don't know. So what we're taking all the compounds, and what you see here is HPLC pattern, and every small dot here, it's a different material, terpenoids, flavonoids, or cannabinoids. And what we are doing, we're saying, okay, we have 100 compounds in this extract. Let's divide them to 25, 25, 25, and 25. So we're doing fractionation with the equipment that we're doing, and fractionate the oil to specific compounds, 25, 25, 25, 25. And we're going back to the cells, and we are very lucky that just 1 piece of this fraction worked. So 3 fractions didn't work at all, and 1 fraction was as good as the whole extract. And we did it in biochemistry and did it with different ways, and we showed that 1 fraction is working. So we already eliminate 75% of the compound. So we're going on, we're taking this fraction and defractionate it again. We're doing another fraction, and we're doing combination and fraction until we succeed to isolate 3 specific phytocannabinoids. Then when you take these 3 of them, they're working as good as the whole extract. So we need just 3 of them, but every combination of 1 of them or 2 of them together is not working as well as the whole 3. The whole 3 is working exactly like the whole extract, but every combination of 1 or 2 of them not working as good. So we have the Entourage effect, but it's not a whole extract, it's 3 compounds. One of these compounds is actually an unknown compound, doesn't have a name. We were the only one that published these compounds 2 years ago, and this specific compound is an unknown compound that we are the first one that discovered them. So we are working from 1 strain. Every 3 months, I'm getting a batch from the grower, creating an extract, treating the mice and treating the cells, and working with blood samples from humans, and sees its effect. A year ago, in 1 day, it stopped working. My student, my PhD student, Eli, come very white, even though he's usually not white at all, and said to me, Dedi, this is not working anymore. I don't know what's happened. We analyze it and we see that this specific molecule is not expressing anymore. I don't know why. They changed the greenhouse, they changed the soil, the sun, it was very hot. I don't know, it's a plant, it can be changed. So we were in a situation that now I don't have any strains that have this combination of these three. So what we did, we went back to our database, we looked which strains have one molecules, at least one of them, this unique one, even though these strains don't have the other two. And we combine two strains. We take a strain with two molecules, a strain with one, and we combine it, it's work the same. Moreover, if we are purifying one compound from here and two compounds from here, 100% purified, we can get the same effect exactly in the mice that we got with the one strain that we have, the three of them. You see how it's curing the leukemia, downsize the tumors, and moreover, when we're taking these tumors from the mice, even though the mice get the cannabis orally, we can find in the tumors the three compounds, the three phytocannabinoids accumulating in these tumors. So even though the mice take it orally and the tumor is now in the back or in our immune cells, we can define these three molecules accumulate in these mice. So what we're doing now, together with the Technion, I opened a company that's raising money to take these results to clinical trials. And I hope that in 4 to 6 months, we will start clinical trials in humans. But here comes an important point. What is clinical trials in cannabis in humans? We actually have two different roads. One road is the classic road, the pharma road. We can take three isolate compounds, do toxicity in humans, and then clinical trials, phase 1, phase 2, FDA approved, 7 years, $500 million, and if we will succeed, I probably will buy the Technion. The other road is the cannabis road, the whole extract. Actually, if I'm taking the whole extract of fraction from the cannabis, I can start clinical trials right away. In Israel, we have 40,000 patients that get cannabis under prescription. 18,000 of them getting cannabis for a cancer treatment, but it's not treating the cancer, it's treating the palliative treatment. It's reducing pain, increase appetite, reduce nausea. So patients that have cancer in Israel that getting chemotherapy or radiation can get immediately cannabis to hold this treatment of palliative care. So actually, I have a patient already in Israel that getting this type of cannabis as a cancer patient. So if I'm taking one strain, this specific strain, or combining two strains, I can start a clinical trial, what we call phase 2B already, in 3 to 4 months, and check that if this really works. And if it's working, in a year, year and a half, up to 2 years, I can have a product that can really treat patients. It's true, it's not the totally pharma isolate compound, but it's a product that maybe can save lives. As we are doing that for cannabis, we can do that for other illnesses. And what I'm showing you now, and I will show that really in 2 minutes, is a system that we developed for multiple sclerosis. Actually, 2 years ago, I visited my friends in Olivia Newton-John Hospital, and a collaboration that we are doing there. And I have few meetings in Marlborough, and I got a call from Sydney, from the Technion Society, if I can come and give a talk. I fly to Sydney and I gave a talk for 2 hours. And one of the people from that talk approached me and said, look, I have a daughter that have a multiple sclerosis and whatever. And we create a collaboration with this group, and we are working with them. It's one of the companies, the Australian companies, and we're doing this multiple sclerosis test with cannabis. And what you see here, we're taking a mice, we inject the specific materials, and we develop a disease, the multiple sclerosis. And you can see the development of the disease. If as it's higher, the meaning that there is more a clinical score of the disease. And around day 20, the mice is extremely sick, and you can measure how much he's crawling his legs and other parameters. and around day 30 be curing from this disease. But on day around 20, the disease is more severe. So we're taking pure CBD, we're giving to the mice pure CBD, and we see some of improvement of the disease. But now we're taking high CBD strains, 2 high CBD strains, and we see much better improvement. So on top of the CBD, there are other compounds that helping to improve the disease, as we saw in the leukemia. But moreover, we're taking a different high CBD strain, the same amount of CBD, and it's making the disease worse. So we have one high CBD strain that make the disease, improving the clinical score of the disease, and other strain with the same amount of CBD that make it worse. And what we're doing from here, we're starting to define which molecules are important to improve the disease and which molecules can be dangerous and shouldn't be given. The same method that we did with the leukemia, and we're doing this for Alzheimer, for sleep disorders, and other epilepsy and other illnesses. This approach is not limited to cannabis. Actually, my lab today have all the equipment to analyze small molecules from plant, to do extraction. We have ethanol extraction, butane extraction, CO2 supercritical, press. We have all this equipment to take a plant, to dissect it to the compounds, to understand what it's doing, and to take it to different levels. So for an example, I have a student that collect 96 different algae from the sea, the Israeli Mediterranean Sea. We did extraction to all of these algae. We screened them on few diseases. We defined few algae that affecting them. And this was a start for 2 PhD work in my lab. Cannabis in general opened the door for a new approach of medicine, which is more complicated medicines. Few years ago, we look on medicine as single compound, single target. We take in salicylic acid from for willing widow, and we extract it, and we learn that it's affecting our veins, and we call it aspirin or different way. But we're taking from a plant one molecule, we create a medicine that's doing specific effect, and we're using that. Around 80% of the medicine out there are specific compounds that take from specific plant. Even chemotherapy, Taxol is from Taxus, vincristine, vinblastine, they are from vinca. Many chemotherapies and various treatment, it's one molecule from one plant. But if we're going back to medicine 100 years ago, 150 years ago, you will go to the bushman or to your medicine guy in the village, and you have kind of a disease, you will pick this leaf, take this flower, take this root, will combine them together and give you a medicine. It was a more complicated medicine. And we are approaching that today. We understand that cannabis in one plant holding a lot of compounds that doing complex actions. But if you're going back to medicine, it was a combination of things, and plants are more complex. The medicine and physician in the last 10 years understand this complexity and started to go that road. One of the major medicines that we know is the cocktail that HIV patient is getting. It's not one medicine, it's a cocktail of compounds that being given to HIV. It actually saved millions of people in the world from this virus. We know in cancer that today, the physician a lot of times give a combination of chemotherapy, a cocktail. They give it Taxol, vincristine, and tamoxifen together, because every one of them attack different pathway. And I think the cannabis opened the door for a plant medicine that was hidden because it was too complex. And today we can do that from algae. There is a big movement to start to check mushrooms. And I know at least few companies in Sydney and other places in Australia that started this. Some of them are mushrooms that have been known for years to be used as a medicine, and it started to move. And my lab is surely taking this approach too. In this complex medicine, we can do things that are very accurate also. We can take the whole extract, and this is from a publication that we published last week about, we took the whole cannabis extract and we put them in nanobeads. Now, these nanobeads, this polymer, hold this extract and release it slowly, slowly. So if you take this one and we inject it into the mice under the skin, this whole extract that we know that reduced seizures in mice was released slowly, slowly. In order to give every day the mice cannabis, we can inject one time every 2 weeks. And actually we saw that it's working better because it sustained the amount of these phytocannabinoids in the blood in the same level. We're talking about whole extract, complex, a variety of cannabinoids, but still accurate in nanobeads that can be injected under the skin, release it in a slow release, and in 2 weeks, release always the same amount, sustain the amount, and we see that it's actually working better than the extract that been in the same extract that in these beads. This is just an example how we can improve a medicine even if we are staying in the whole extract. I want to finish my short talk here with saying a big thanks to my amazing team in the lab that working and doing all this amazing work. I showed results from Dr. Gil Levitus, who is the head of my neuro department, the multiple sclerosis. He and he's doing the main work. I showed the cancer work being done by Dr. Liran Baram and Dr. Eli that did all this work. I also want to say a big thanks to my company. I have a company sitting in Caesarea, Caesarea, which doing the most of the complex analytical isolation and extraction and all of this, and part of the work that I showed being done in Canasol. I want to say that 5 years ago when I started to do that, I have 5 students in my lab and we were extremely passionate to do this work. When I'm looking 5 years after, today I have 43 students in the lab and 38 people working in Canasol. So altogether we are a group of around 80 scientists that are doing this work, and this is extremely exciting period and extremely exciting work. And I want to thank you very much for listening till now. And Adam, I'm free to answer any question that you or the audience have.
Okay, Dedi, thank you. As always, it is highly entertaining and informative to see you talk about the work you're doing in the space. You know, it's so important that the mystery of cannabis is unraveled and we have a better understanding of how the cannabinoids, whether isolated or together, are able to treat various therapeutic applications. So I think the work you're doing is absolutely vital for the development of this space. So I had a couple of questions for you from the audience and a few that I've prepared. Something that I note that you didn't speak about on this presentation that I'd love you to give a bit of context to is the work that you're doing, your database project. So are you able to talk a little bit about the work that you're doing with growers and patients in conjunction with one another and how you're combining all that data?
Sure. 4 years ago, I got a big academic grant from the Evelyn Liefer Foundation from United States to do a database. And what we proposed and what we're doing in the last 4 years, we said, actually, we know exactly what the patients are getting from bottom to top. We can analyze all the cannabis they are getting in Israel because we have just 8 authorized growers that supply around 90 different products to the patient. We create a database that every time that one of the growers produce medicine, produce cannabis to the patient, every batch is first coming to our lab with doing full identification on the molecules And we have a fingerprint and we have a database on all the cannabis materials that the patient can get in the last 4 years. On the other hand, we started to follow up on the patient. We built a questionnaire online for the patient that they are filling it once a month. And we're trying to combine these two together. It said, okay, this is the materials that the patient is getting. This is the output. He can sleep better, he can eat better, it's affecting his pain, whatever. And we're trying to combine this in a big statistical and artificial intelligence work to understand which strains and which compound affect which disease and which effect. And when we have these results, we're taking it to preclinical and clinical work to try to verify if what we found is working. So when I want to start to work on sleep disorder, I already knew which strain in Israel is affecting sleep in which way, and now we're trying to understand the mechanism and how it's working. So this is the database that we created in Israel.
So from once your team has some sort of statistical significance of, you know, strain A impacting indication B, what's the average time cycle for you to get that into the lab and start testing?
So if I have something very, very significant, I'm usually starting very fast, okay? So we saw there is a mutation called APC in a colon cancer patient, and we saw that usually patient, it's a genetic mutation, and usually when a kid have this mutation, in age 10 to 12, he will start to have a polyps in his colon, and it will grow and grow and grow until he will develop a cancer, or you will need to reduce his colon. And we realized from the hospital and from the database that patient, even though it's kid that's using cannabis, is actually blocking the polyps to arise, and it's reducing them. And we have a very solid data about that. And so immediately we started to do a research. We developed a animal model, like a mice model with this mutation that creating these polyps, and we create organoids from this, and we are doing this research to understand which type of cannabis, which molecules, and what is the mechanism that affecting this disease. But this is happening quite fast. When we have a result, when we understand the mechanism and the molecule, this takes years.
Okay, that's fair enough. I mean, that's the most exciting and frustrating part about this space, is that the investment of time is significant. The outcomes are potentially enormous. A question that I like that one of the audience put forward was around, I'll kind of put into my own question. What are the three cannabinoids you're most excited about right now, and why?
So, you know, it's kind of asking me, I have 4 kids, which one I love more, right? It's not working that way. I don't have a specific cannabinoid that I'm excited about. I think there is a bunch of cannabinoids which are unknown, the 3318, 3319, and whatever, that we see that they kind of highly expressed in the plant. And I think they will be found very important. I think the V family, THCV, CBDV, are extremely important, and there is a lot of efforts of finding them. I have already results with CBDV that's important to specific illnesses. I'm sure that THCV is very important. We know that THCV is very psychoactive. It's a different psychoactive than the THC. It's a different way of feeling, at least that's what people say. So the meaning that is very effective and binding receptors and activating things. I think the V family will be very important, and I think few unknown cannabinoids will be very important. I think also there is a few flavonoids that are unique to cannabis, cannaflavin A and cannaflavin B, and I think we will quite fast discover that part of what we see and we think it's cannabinoids, it's actually these 2 flavonoids.
One of the questions that's come up several times from participants is, you know, what cannabis strain should I use for my specific illness? There's a number of questions where people have asked, how cannabis can be used to treat various indications. How do you respond to patients when they come to you and say, what strain should I use to treat my, my specific illness? How do you go about addressing that? Because it's a very complex question with not a particularly simple answer.
No, so usually I don't have an answer. First of all, if you are not from Israel, I'm not familiar with the strains and they are very different. So I don't know what is purple kush in Australia when it will be very different from the States. So I don't know what are the plant materials. This is first. Second, if we'll take cancer, there are 100 types of cancers. Cancer, it's a given name to 100 of different diseases. We're talking about breast cancer. There is more than 40 types of breast cancer. And every cell in our body will have its own types of cancer. So we have around we have few hundreds types of cancer. If we're taking cannabis, just in my lab, I have 900 different varieties of cannabis. If each one of them have different combination. So it's more than a lottery to define which cannabis can affect your cancer. And if you ask me, I don't believe that cannabis affects any types of cancer. It will affect specific types of cancer. Moreover, cannabis affecting ourselves mainly through our endocannabinoid system. And usually when people are talking about endocannabinoid system, they are talking about two endocannabinoids, anandamide, 2-AG, kind of 2 or 3 receptors, CB1, CB2, maybe GPR55. In the blood samples or a tissue, I can analyze 150 endocannabinoids and around 37 to 39 receptors. So also this is very complicated. Which receptors expressing which types of cancer will be important if we can affect these cells or not, and which types of cannabis. We are trying to develop that. I have in my company, I have a subgroup that doing all the receptors. We call this company 7 Helix, trying to analyze which receptors expressing on which types of tumors and how these being affected by cannabis. But I need more time. For now, usually I will tell to the patient, go with the regular treatment. The success, the rate success is still much higher than cannabis. Cannabis is still a lottery in these types of diseases. So this is when I'm talking about cancer. When we're talking about sleep, pain, appetite, epilepsy, there the rate of success is bigger. And my answer is, why not to try something that have less side effect and can be useful. So, but on terminal disease like cancer, I would still be very, I wouldn't use cannabis yet until we'll have better results.
On the note of cancer, one of the areas that I'm particularly fascinated about your research is related to the cannabis's ability to create or to allow for cancer cells to commit cell suicide. Can you expand a little bit about that and talk about how cannabis actually impacts cancer cells?
Yes, so first of all, in every type of cells, the mechanism is different. But what's happened, cancer, it's a given name to 100 different diseases, and we put them in the box. We said every time that we have 1, 2, 3, 4, 5, 6 hallmarks, we will call it cancer. One of the first things that happen in our cell, there is a mechanism of committing suicide. When cells understand or absorb that there is a major defect in the cells, he will commit suicide. It's called apoptosis. What's happened in cancer cells, which was normal cells, they had kind of a block. They are not committing suicide. Even though there is a major defect or major mutation or whatever, they are not committing suicide. What we see in cannabis, that it's actually bring back the ability to the cells to commit this suicide, to create apoptosis. So if we're taking the leukemia cells, we realize that the cannabis is blocking specific protein on the Golgi apparatus that don't let the protein to go to the membrane. It's going to degradation, and that reason move the red line to the cell, said, okay, this is too much, I will commit suicide, okay? But it can be different in different types of cancer. Let's say in breast cancer, it can block the the estrogen receptor to move from here and there, and then the cell will commit suicide. And in every type of cancer, it's a different mechanism, but in general, what it's doing, it's moving the red line. The cells have a red line, you know, and the cancer cell said, oh, this problem, I'm still not committing suicide. You move this red line and said, okay, this is too much. Now I will kill myself. It's not far from what doing chemotherapies like Taxol that block the microtubules depolarization and the cells committing suicide, or other mechanism of chemotherapies. The difference that usually cannabis is not killing normal cells. And this is the unique thing here. What, you know, chemotherapy is killing every dividing cell. We are looking on more specific, it's the medicines, not just cannabis. We're trying in the last few years to do more personal work, that it's our medicines will kill just the specific cancer cells. And there is other drugs today, like Herceptin or whatever, they're doing a very chirurgic work on cancer cells, and this is the way the oncology is looking now to treat cancer, and cannabis is part of that.
We've got time for one last question, and what I'm going to ask is around where you see the cannabis industry in 5 years from now. If you've got a crystal ball, considering you're at the cutting edge of research, where do you think this is all going? Where do you see this in 5 years?
So first of all, I think it's kind of clear that cannabis won't disappear. There are enough illnesses that we know that it's working, and you can't take the wheel back. If we'll take epilepsy, in Israel we have around 1,600 kids with severe epilepsy that getting cannabis that's reducing this epilepsy. But in Israel, as a child, in order to get cannabis for epilepsy, you first need to try at least 5 different medicines for epilepsy and to be resistant on that. So these are kids that try all the medicine, resistance to all the medicine, and actually we don't have a treatment for that. Usually these kind of kids with a severe epilepsy won't survive. They will reach age 12, 14 and will die. So these kids now getting cannabis, and it's reducing the seizure 50%, 70%, and we have also few kids which are seizure-free. Think about a child that have major epilepsy, probably wouldn't survive, now getting cannabis and seizure-free. There is no way that you can take the wheel back and take it from him. This will save, okay? The question, how we will see this medicine on the shelf, okay? And I believe that we will see 2 types of medicines that coming from cannabis. One type of medicine will be very accurate. 2 molecules synthetic from the plant that the pharma company produce and sell as a pill or something like that, and you taking this will help you for sleep. This will reduce, seizure, something like that. This is one part. But the other part is more holistic, the whole extract. If we're looking on pain patient, PTSD patient, autistic kids, cancer patients that take cannabis for palliative care, they're taking the cannabis as a holistic treatment. It's reducing a little bit pain, increase appetite, improve the mood, help with sleep. It's doing many things together. Instead of taking 7 pills, you're taking cannabis, very mild, but a little bit here, a little bit here, a little bit here, a little bit here, and you're improving the whole system. You're improving the quality of life of the patient. And on that type, I think we will have cannabis medicines that will be for quality of life, that helping for pain patient, PTSD, epilepsy. On that term, we won't be able to purify specific compound. It will be a complex of compounds that each one of them doing different things. But we will see very accurate products. It will have a name, it will have branding, there will be a lot of commercials that will say, our product is working better, it will be very stable, it will contain always the same compounds in very accurate way, that every time that you will take, you know what is the dose that you need to take, and what is working for you in which way. It will be accurate, but still whole extract, something like that. This is my vision here.
I've got so many more questions for you, Dedi. However, we are now at the wrap-up point, and I want to bring Jono online to close out the discussion. Dedi, always a pleasure. It's really
Adam,
lovely to
thank
hear
you
you,
very much.
see you, and to interact. So thank you very much. Jono, I'm going to pass it over to you now, mate.
Wonderful.
So just before you are talking, I want to say again, thank you very much for inviting me. Thank you very much, Adam, for hosting me. Always pleasure to speak with you and to see you. And thank you very much for the audience that listening to us. It's a pleasure to be here and to speak. And thank you very much.
Well, look, I'd add the thanks. Dedi, we see behind you the sun is shining brightly, and we know it's super hot where you are. Just want to say the future looks super hot when there is just, I'm not technical, but it sounds in the number of oids, cannabinoids, and all the oids, it looks like there is an amazing treasure trove that you have, and if you find the right one, you have the keys to the kingdom. And you know, wish you well in that struggle. I just wanted to say, behind me you'll see my banner, North Sydney Innovation Network, and for our part, we educate well, as it says, network, educate, and advocate for startups and tech on the North Shore of Sydney. So we love being with Technion and with Ori, because part of that, the middle word is educate, and we love bringing the education from around the world to the people looking and listening today. Thank you, Ori. Over to you.
Thanks, Jono. So just final remarks. Again, I wanted to thank Jono from North Sydney Innovation Network and Ron from the Embassy in Canberra for supporting us once again. There's a lot happening at the Technion. As for those of you who are not aware, there's research all the time, including now relating to COVID-19. And if you're interested to find out more, like I try to kind of add to the chat, please sign up to our newsletter or give me or send me an email to follow up. We'll be very happy to talk and tell you more about what's happening. I'm sure that we could have stayed much longer, and looking at the amount of questions that piled up, we could have been here till midnight, Israel time. But unfortunately, Dedi has to leave. But what I will try to do is take some of those questions offline and see if you know, if Dedi would be keen to answer, and we'll send it out in our next newsletter so you can stay informed. We've got a lot of events coming up, so make sure you tune in and stay up to date. Again, Adam, thank you so much for giving up your time and your knowledge and sharing it with us today. Dedi, of course, it has been a privilege having you, and we look forward to welcoming you in Australia once again, once there's flights available as a starting point. Until next time, keep well, stay safe, and we'll see you again soon. Take care. Bye-bye.
Thank you very much. Bye-bye.
Bye.
Take care, everyone.